Hello, I'm Kazuhide Asakawa from Tokyo Medical University. So first of all, I'd like to thank the organizers for giving us this wonderful opportunity to present our recent work. So here we are looking at 6 zebrafish embryos, and if you look closely, you will notice these are top 3 embryos responding to blue light. And the trick I'm using is that these embryos are expressing the light-gated sodium channel called channel rhodopsin in their spinal motor neurons. So I brought this movie to convince you that zebrafish is very friendly to optogenetics, and you can not only visualize but also manipulate spinal motor neurons in intact animal. And in this presentation, we would like to apply zebrafish optogenetics to study the function of TDP-43 in the spinal motor neurons. In ALS, degenerating neurons are characterized by cytoplasmic aggregation of a DNA-RNA binding protein called TDP-43. We have been interested in the mechanisms how the normal TDP-43 in the nucleus turns into these pathological aggregates in the cytoplasm, and trying to understand the mechanisms that generate TDP-43 cytotoxicity causing neurodegeneration. In healthy cells, TDP-43 is known to regulate chromatin structure, transcription, and various steps of RNA processing, DNA damage repair in the nucleus. And in the cytoplasm, it also regulates RNA processing, transport, and translation. It also controls mitochondrial function and calcium homeostasis. So we can easily imagine that these TDP-43 functions are compromised in ALS motor neurons, where nuclear TDP-43 is lost and deposited as cytoplasmic aggregates. But what we currently don't understand is whether the cytoplasmic aggregates themselves are the cause of cytotoxicity and degeneration, or cytoplasmic aggregates are just byproduct of neurodegeneration, and TDP-43 toxicity stems from some other facets of TDP-43 abnormality. Addressing this question is important not only in understanding disease mechanisms, but also in determining whether we should consider the cytoplasmic aggregates as a therapeutic target. So to address the question, in this study, we developed an optogenetic TDP-43, which we named OP-TDP-43. So in OP-TDP-43, RFP is tagged to the full-length TDP-43, and the crypto-olig tag is fused to the intrinsically disordered region, or IDR, of TDP-43. So crypto-olig is a domain of Arabidopsis cryptochrome 2 carrying a mutation that enhances light-dependent clustering. And the idea is that blue light illumination enhances crypto-olig interactions, which in turn promotes IDR interaction and phase transition of TDP-43. So for OP-TDP-43 to be photoswitchable TDP-43, it has to be functional as TDP-43 under dark condition. So we found that OP-TDP-43 is indeed functional because it rescued blood circulation defect of a TDP-43 double knockout fish as efficiently as wild-type TDP-43. So next, to test whether OP-TDP-43 forms cytoplasmic aggregates by blue light illumination, we first looked at the skeletal muscle because myofibers have large nucleus and large cytoplasm, So here we are looking at the lateral side of the body, and you see OP-TDP-43 enriched in the nuclei, but not in the cytoplasm. So now when you shine blue light, so I hope you can see that OP-TDP-43 gradually appears in the cytoplasm as granular structure or droplets. And consistent with the fact that TDP-43 is a component of ubiquitin-positive inclusions in ALS. So these OP-TDP-43 droplets are partially ubiquitin-positive. And we found that this OP-TDP-43 droplet formation was much slower than cryo-oligo clustering. As shown here, OP-TDP-43 droplets become evident about after 2 hours of illumination. But under the same condition, cryo-oligo on its own form aggregates as soon as you shine the blue light. So what we are speculating is that TDP-43 modules inhibiting cryo-oligo module from developing into large aggregates, and OP-TDP-43 forms droplets by TDP-43 dependent mechanism. And also we would like to mention that we have done the same set of experiments in the epithelial cells and matured myofiber, but we didn't see light-dependent OP-TDP-43 droplet formation, suggesting that the cytoplasmic droplet formation is the cell type dependent phenomenon. So then, what happens in the motor neuron? By using a GAL4 driver for motor neuron, we expressed OP-TDP-43 in almost all spinal motor neurons, and at the same time visualized soma with EGFP. So here you are looking at a spinal cord from the dorsal side. You can probably see that OP-TDP-43 in the nuclei is slightly smaller than the soma visualized by EGFP. And if you shine the blue light, after 4.5 hour illumination, we found that OP-TDP-43 signal and EGFP signal almost overlap, suggesting that OP-TDP-43 is dispersed throughout the cell. So if you closely look at the single cell over time, OP-TDP-43 in the nucleus is dispersed throughout the cell after the illumination and overlaps with EGFP signal. And interestingly, OP-TDP-43 is enriched again in the nucleus after illumination is stopped. So here we can quantify cytoplasmic mislocalization by dividing cytoplasmic signal by nuclear signal. The cytoplasmic mislocalization index transiently rises by the light illumination. So we are surprised to see that a prominent cytoplasmic mislocalization of OP-TDP-43 rather than droplet formation. So next, we tested whether this transient mislocalization is toxic or not. For that purpose, we employed GAL4 driver for specific motor neuron type. Zebrafish has about 60 spinal motor neurons in a spinal hemisegment. And this PRDM14 GAL4 driver labels unique motor neuron, which is a cap motor neuron. So you can clearly visualize the axon arborization and measure its total length. And we found that A transient 3-hour blue light illumination was sufficient to perturb axon outgrowth of cap like this, suggesting that OP-TDP-43 oligomerization has a dominant negative effect. To explore the effect of transient OP-TDP-43 mislocalization on already established neuromuscular junctions, we focused on the later stage where motor axons are more arborized, and the collateral called DCCT is formed. Before light stimulation, OP-TDP-43 expression itself didn't affect total length of DCCT. But after the illumination, the growth rate of axon was significantly affected in the OP-TDP-43 expressing neurons. And I hope you can see that some neurons show a growth rate below 0, which means that axon shrinked. To test whether this axon shrinkage involved denervation, we employed live imaging system for neuromuscular synapses that we previously established. So by visualizing presynaptic protein and acetylcholine receptors, we found that after the blue light illumination, some of the neuromuscular junctions are disappeared, which is indicated by yellow arrows. So these observations show that light-dependent oligomerization of OP-TDP-43 promotes myofiber denervation. So, why didn't we observe OP-TDP-43 aggregation in the motor neurons? So far, we used confocal laser illumination against agarose-embedded fish, and we restricted illumination duration from 3 to 4 hours to maintain fish viability. So then, to extend the illumination duration, we set up the arena where freely swimming fish can be illuminated by blue LED light. So using this system, we found that OP-TDP-43 formed cytoplasmic droplets, and these droplets developed into distinct cytoplasmic aggregates after 3 days of illumination. And if you co-express non-optogenetic EGFP TDP-43, it also is recruited to the OP-TDP-43 aggregates, showing that OP-TDP-43 sees TDP-43 aggregation. And in this condition, fish are viable and displayed no obvious locomotor deficits. But we found that when we introduced ALS-causing mutation in IDR, which is A315T mutation, we saw similar seeding activity, but in this case, we observed locomotor deficits like this, suggesting that IDR is involved in toxicity of OP-TDP-43. So let me show the example of locomotor deficit. This is the healthy sibling, and this is the fish expressing OP-TDP-43 carrying ALS mutation A315T and illuminated for 3 days. So this sibling is swimming and floating, so it's off focus. And this ALS fish is lying at the bottom of the dish. And it has normal heartbeat, so it is actually living but cannot move. So let me summarize this talk. Under physiological condition, IDR-mediated TDP-43 oligomerization is rarely observed or quickly removed by proteolysis or nuclear import pathway, and we don't observe cytoplasmic aggregates. On the other hand, we learned that optogenetic oligomerization significantly increased the cytoplasmic pool of TDP-43. And we found before TDP-43 aggregates are deposited, axon shrinkage and myofiber denervation occurred. And we also found that long-term illumination generated cytoplasmic OP-TDP-43 aggregates and recruited non-optogenetic TDP-43 as well. So although we didn't see nuclear depletion in this study, much longer stimulation might be able to cause nuclear loss of TDP-43. So in summary, we would like to propose that TDP-43 oligomerization triggers both acute and long-term pathologies in the motor neurons. So coming back to the original question, is cytoplasmic TDP-43 aggregates cause cytotoxicity or just byproduct of neurodegeneration? So we realized that our system cannot answer this question because we found that oligomerization-dependent cytoplasmic mislocalization is already cytotoxic. So instead, this study predicts that cellular factors that promote oligomerization-dependent cytoplasmic mislocalization may be important therapeutic targets to protect motor neurons from degeneration. So I would like to thank Dr. Koichi Kawakami and a lab member at National Institute of Genetics. And I would also like to thank Dr. Hiroshi Handa and lab members at Tokyo Medical University for their support. And I would like to thank also funding sources for their generous supports. And please take a look at this paper if you would like to know further details. And thank you very much for your attention.